Good afternoon, everyone. Is my audio clear?
Yes, sir.
Okay, thanks. So in the last class, we were discussing about some fundamentals of developmental biology or embryology, you can say. And we will continue with that today as well. And then we will read some more of the advancements which we may have read in other courses. So in the last class, we discussed about zygote, the morula, blastula, gastrula stage, fetus. And in each of these cases, we discussed about what is the important thing. And in that, we discussed the fertilization, and in morula, undifferentiated a ball of or a sphere of cells, identical cells. Blastula, the first differentiation and the formation of the inner cell mass and trophoblast. In gastrula, we discussed about the formation of primordial germ layers, endoderm, mesoderm, and ectoderm, or the reverse order. And in fetus, the organogenesis and development of tissues and so on. We also have discussed about adolescence or puberty or sexual maturity, and from which on the individual becomes more sexually, mature or capacity to bear children, right? And so on. And eventually we have discussed about spermatogenesis and oogenesis. That's where we stopped. I'll have a quick review of where we stopped and some of the amazing things about oogenesis or spermatogenesis. We'll see it in different ways. Primordial germ cells and primordial germ cell here. They are the ones responsible for the germ plasm of the body. Primordial germ layers, that is ectoderm, mesoderm, and endoderm, are all responsible for the somatoplasm. And primordial germ cells are responsible for the gametes or the germ plasm. So we are discussing that right now. And spermatogonial stem cells, in first difference you can notice is there are 4 sperm cells that are formed upon meiosis of one spermatogonium, or primary spermatocyte. And whereas in oogenesis there is only one egg cell that has formed. The others are excreted or expelled as first polar body and second polar body. The egg, it's kind of one nucleus steals all the cytoplasm from all the rest of the cells. And it is highly nutritious with lot of loaded food materials that were required for further cell divisions upon fertilization. The other one is the meiosis in spermatogenesis in the male. It happens after puberty. That is at the age of around 15 or 16 years old and so on. Whereas in female, the meiosis has already started even when the baby is at the embryo stage itself, and is arrested in the middle of the meiosis or prophase of meiosis 1. It's something like it's arrested between these stages, where completion of meiosis 1 and onset of meiosis 2. Upon puberty, an egg cell that is ovulation occurs, and then the egg cell is released. We will see in the next slides. And then upon fertilization, the meiosis is completed. Okay, and the spermatogonium or primary spermatocytes can be produced until the age of 95, or actually it's there is no limit. Okay, whereas in females there is a limit, maybe around 45 or between 45 and 50 years they undergo menopause, which means that there are no more production of the eggs, or ovulation stops. Then the menstrual cycles are also not there anymore. So they're highly controlled by hormones and so on. So in today's class, we will discuss a little more and try to focus with some details which may not be, you don't have to memorize, but you should be able to appreciate what the biology is and try to know some details of it. So there are two gonads or the ovaries in a female body, and one on the left side and the other on the right side. And the egg, the ovaries are the place where all these ova are present, which are supposedly already made during the embryo stage, and they remain dormant until the puberty or until the ovulation of the day. Ovulation starts only after puberty. So either of the gametes, one is on this side and one is here, either of these gametes will be active in any menstrual cycle. So only one follicle will mature and releases egg. And that process is called as ovulation. And then the secondary oocyte keeps traveling along the oviduct or fallopian tube. And if fertilization occurs, this is somewhere here. The sperm is deposited during copulation, and the sperm will have to travel all this way and meet the egg around in this place, and fertilization would occur there. So upon fertilization, then now you have the formation of the zygote, and then the first cleavage refers to here in this case is about cell division. One cell divides into two. And this cell division continues, and the morula forms, and then at one stage it forms the blastocyst, or around that time it gets impregnated into the endometrium. Endometrium is the lining of this uterus, which is supplied with a lot of blood, and that blood brings nutrition and oxygen and many other things that are responsible for supplying nutrition to the growing embryo. Right? During menstruation, say for example, if this egg cell is not fertilized, then it would not impregnate, and then this endometrium along with the egg would have been discharged as menstruation. That's the common biology, and we will learn about it little more details, and then in the next chapter and so on. This is just a introductory chapter to bring you all or familiarize you with all the terms and so on. Okay. So we will discuss in today's class, mostly we are going to discuss about the process of fertilization and the complexities involved in that. So if you see and while you are listening through, please, as I said last time, please take a paper, try to write down some of the words that you might have come across for the first time, or the words for which you don't know the meaning of. You don't need to write the meaning, just familiarize yourselves with those words. So zona pellucida is the thick lining that is present around the egg cell. And here is granulosa cells that may be some more cells from the ovary that might be still attached to the egg cell. And here is the first polar body. Its genomic content would typically be about 2N. And this is the nucleus, which is about to undergo meiosis 2. And it has N's and N. This side is 1N, and this is another N. Right, so you have 4N, which before undergoing meiosis, I hope you remember the cell cycle. If I didn't mention, you have G1 phase, there is S phase, there is G2 phase, and then you have meiosis or mitosis. G1 phase, if you take it as humans, for example, you have diploid state, and during S phase, the DNA is replicated, so the 2N becomes 4N, It's somewhere around 92 chromosomes. And during mitosis, if it is mitosis, you will have 2 cells, each consisting of 46 cells. If it is meiosis, we will have 4 cells, each consisting of 23 chromosomes, right? So duplication of the genome happens in S phase. So if you, what we are going to, I hope you remember the zona pellucida, and there is the cell membrane of the egg, and you have the zona pellucida here. And at any given time, one would only want one sperm cell to go in. We don't want multiple of them to go. And that is where we want to see how is polyspermy inhibited. What is polyspermy? Entry of multiple sperms into the egg cell, which is not good, because we would always want that the number remains 2n. We do not want 3n or 4n, because the cells will die if anything happens, or there will be a lot of abnormalities. So some of the things that happen during fertilization are something like this. So before we get in, we will talk about also the head, the nucleus of the sperm. The head region contains the nucleus of the sperm. The tip of it has a sac-like structure, which is referred to as acrosome, that consists of so many proteolytic and other enzymes that can dissolve the zona pellucida. So using that, it digests through the layers and tries to reach the cell membrane of the egg. I hope you remember, or you might have heard, that out of billions of sperms, you are the one who made it to the egg, right? So act like one. That is how some motivational speakers do speak. It could also be that in reality, there could be multiple sperms working to degrade and fall out, and there may be some other sperm that might be taking the same route and falling off, and eventually some lucky sperm actually gets to fertilize the egg, right? So we don't know if it is the strongest one or if it is the luckiest one that is actually fertilizing the egg. I think there is something, a term called as success paradox. Not necessarily that everybody who worked hard would succeed. There is also timing. There is also many factors, including luck. I think you may want to find it out just for knowledge's sake, or trying to be philosophical at times. Okay, so the sperm, the objective is to dig in, and somehow whichever one delivers its nucleus into the egg cell through fusion or rupturing of the cell membrane would become a part of it, and eventually will contribute to the formation of the organism. Once one egg cell has gone in, we do not want any other sperms to fertilize it. We don't want any more sperms to fertilize the same egg that's already fertilized. That would be called as polyspermy, where one egg cell is fertilized by multiple sperm cells, and that is going to die. It will not form a fully functional organism. So as soon as there is one fertilization occurs, there are many changes that would happen. There are cortical granules that are secreted into the space between zona pellucida and the cell membrane. So the next sperms will not be able to deliver their nucleus into the cell. Okay, that space between the zona pellucida and the cell membrane is referred to as something like perivitelline space. We'll go into further details and try to see what is happening at the molecular level. When the sperm binds to the egg cell membrane, there are several changes that include sodium influx, and because of that there is a membrane potential typically for every cell, and when it is disturbed there is sodium influx into the cell, because of which the potential has changed, and that potential itself will act as a fast block to polyspermy. Somehow inactivates or prevents. It's not very effective, but for immediately to act, it's a light dose kind of thing, you can understand. Quickest way, but not the most effective way to block polyspermy. The other one is by stimulation of kinase. Kinase is any enzyme that transfers a phosphate group from one molecule to another. And kinase phosphorylates phospholipase C, an enzyme that is embedded in the cell membrane, and that results to production of inositol triphosphate and diacylglycerol. And these two molecules will have further downstream activities, or it's actually a network that we will see here. So here is the phospholipase C, which is activated because of fertilization or membrane disruption. And that phospholipase C will degrade some of the phospholipids that are present in the cell membrane into diacylglycerol and inositol triphosphate, this one. Right? I hope you remember some of it from biochemistry. You have inositol and 3 phosphates, 1, 2, and 3, inositol triphosphate. Diacylglycerol, this is the glycerol moiety, and you have 2 fatty acid chains, so you call it as diacylglycerol. Here is phosphoinositol diphosphate, I think. That is digested into these two. You have 2 phosphates here, P2, and here is another phosphate, and this is the bond somewhere, it breaks here. And this becomes the diacylglycerol, and this becomes the IP3. Okay, so diacylglycerol further activates the sodium proton antiport. So protons are sent out and sodium ions move in, because of which alkalinity increases within the cytoplasm. That stimulates the cell division, DNA synthesis, and many other things that are required for activation of the egg. One should remember that the egg has been inactive for at least 13 years, right? And it has to increase, stimulate its activity to undergo cell divisions and ultimately impregnate into the endometrium and give rise to a full-born new baby. So this also has one more aspect that happens through inositol triphosphate, is that inositol phosphate, I think we should not call it triphosphate. But anyway, IP3 binds to one of the channels, calcium channels, because of which the channel is present on the smooth endoplasmic reticulum that stores that is a reservoir of calcium ions. And the calcium ions now will flow into the cytoplasm, and they activate many things, including cortical granule exocytosis. The cortical granules are moved from here to the between the zona pellucida and the cell membrane, and forming into the perivitelline space, and contributes to the activation of the egg. Here is a nice picture that I show it every year, and here is the point of stimulation, you can think of it, or fertilization, and the sperm entry you can think of, and then you see the red ones that is showing here is the wave of calcium ions that are spreading throughout the cell membrane. And that is how the activity also, that is how the stimulation of the activity happens. It is a very rapid thing, it happens very fast. So the calcium release that we discussed about, which is a product, which is a consequence because of IP3 binding to the calcium channels on the smooth endoplasmic reticulum, and that increase that allows or contributes to the degradation of cyclin and inactivation of MAP kinases, and because of which the mitotic cell division is restored and activation of NADK kinase and so on, membrane biosynthesis happens, and cortical granule exocytosis, formation of hyaline layer, and this is slow block to polyspermy. So these two are very effective. They form later than the previous, that is fast block to polyspermy, but that's not very effective. But this is very effective. And the intracellular increase in the intracellular pH, that is the alkaline conditions, would increase majority of the metabolism in the cell. And what was dormant for so long will actually now become activated, and then will start dividing and forming an embryo. So here is the egg cell, and it is being attacked by multiple sperms, and one of it gets to deliver its nucleus alone. Here it is shown as this. This is wrong. Only the head particle is delivered into the cell membrane. And here you are seeing the first polar body and the second polar body. In this case, you had only the first polar body. Later, upon fertilization, cell division, the meiosis 2, that was in which the egg cell is completed, and then the nucleus 1 from the male, we'll put it as this, and the other one from the female, that is the egg one. They are, prior to merging, they are referred to as pronucleus. Pronucleus. Or if it is plural, it would be pronuclei. And they would fuse, and then they form together, they would form a nucleus. And that's when a zygote is complete. So the 3 is zona pellucida. You can, this perivitelline space, you have polar bodies, and you can think this as the zygote. And now, because of calcium, because of the membrane disruption and calcium ions and so on, you have the egg is activated, metabolism of the egg is activated, and it undergoes first cleavage or the first cell division. Then it undergoes second cleavage or cell division, and then it forms a morula. Morula is a solid sphere of identical cells, right? And from morula stage, the next stage is called the blastocyst. This is where the first differentiation would have happened. The first differentiation is, when you say differentiation, I will repeat what I mean, because it's an important concept that you should remember. One cell has given rise to or has remained like in the same properties, physical, morphological, and biological functions, say, then they are undifferentiated. But if the same cell, because through cell division or maybe because of external cues or internal cues, it could be, and if it has changed its shape and functions, then you say it has differentiated. All these stages, in first cleavage, second cleavage, morula stage, they all look like the zygote. So no differentiation has happened until then. The size might have been smaller, but otherwise the biological functions, everything, and morphology remain the same. But whereas in the blastocyst stage, you would see that there are 2 different types of cells. One is the inner cell mass, the blue ones that are shown here, and the other one is the outer ring that forms. And there is one other difference between morula and blastocyst. It is, you see this void, all this gap that you see, which is referred to as blastocele. In morula, it is a solid sphere of identical cells. Whereas in blastocyst, you see some gap that is happening, that is there, which is referred to as blastocele. So you need to remember ICM and the trophoblast cells, and that stage is blastocyst. So And this is going to be repetition. And one more issue that I want you to remember is that embryology of different organisms, there could be some differences. It's not necessary that it will be all of them are the same. Okay, so try to bear that in mind, but we are taking a general approach to get familiarized with the common concepts in the embryology. So if you see that of a frog or so, fertilization, and then upon cleavage, you have, this is the blastocyst, and here you have the ICM, and the outside is the trophoblast. And the next stage is called as gastrula, or in which gastrulation occurs. That is where the primordial germ layers form. It's called as ectoderm, mesoderm, and endoderm. Okay, so I also want to take a step back and go to fertilization. The whole human body has come from the zygote, right? So therefore, we also refer to it as totipotent. All of the human body comes from ICM, inner cell mass. The trophoblast does not contribute much to the body tissues that we have at this time point. Even more, if you want to go, the primordial germ layers, are the ones that contribute to the formation of all of the somatoplasm. Okay, all the tissues have come from these ectoderm, mesoderm, endoderm, the ones after birth I'm talking about. And then after this stage can be referred to as gastrula, and the next stage is called as neurula, where neurulation happens. That is where the nervous system starts to form, and eventually the whole other organ seems to be forming. Okay, let's have a closer look at what happens in the formation during gastrulation. In humans, the first thing that happens is from the ICM will arrange itself into two layers. One is called as epiblast, and the other one is called as hypoblast. Epiblast is on the top, and the hypoblast is on the bottom. And that is referred to as bilaminar germ disk. So this is also let me go to the next small picture that is attached. I hope you're going to see this. The epiblast that is on the top is going to invaginate like this, and that is called as the primitive groove. And it will pour in some cells that you see that ones here. Those are going to spread around, and that is going to form the endoderm. Here you see that all the yellow cells have moved. So the cells of the hypoblast kind of are taken over or pushed away and is occupied by the endoderm. And between the epiblast and the endoderm, there are some more cells will seep in between them. And that is where now you have the endoderm, that is these yellow cells, and mesoderm that forms between the epiblast, what was an epiblast before, and the endoderm. And that would likely become the ectoderm. The top one, the ectoderm, mesoderm, and endoderm. So this is kind of conclusion to basic embryology, and it's a start of stem cell biology. So and the whole objective is that you pick up the words or the sentences. Fertilized egg will give rise to blastocyst, and all this side, that is one that is showing, that the one that are shown is contributing to the embryonic tissues. All the other cells, like the trophoblast and many things, that hypoblast, they are contributing to extra embryonic tissues. That means it could include placenta and many other things as well. Okay, but they do not become direct part of our own body when the newborn baby is born. So at the blastula stage, you have trophoblast cells and inner cell mass. And then in the gastrula stage, you have the formation of epiblast and hypoblast. And the epiblast, the hypoblast is pushed around, and then epiblast will give rise to amniotic ectoderm and primitive ectoderm. Here we are, okay? And from the primitive ectoderm, you'll have the primary streak. That is where the invagination of the ectoderm happens. And the invaginated ones first forms the endoderm, and then it also between the endoderm and ectoderm, it pours in some more different types of cells that are referred to as the mesoderm. These three are the primordial germ layers. Mesoderm also contributes some parts from it to extraembryonic tissues. Okay, so that's what you should keep in mind. So the other tissues that I didn't mention, trophoblast could form cytotrophoblast and syncytial trophoblast and extraembryonic endoderm and so on. Eventually, the hypoblast will produce the yolk sac. So if we are almost closing up. So the zygote, blastula, and gastrula. We read those and we came to the primordial germ layers, ectoderm, mesoderm, and endoderm. And eventually, there are also from the extraembryonic tissues, there will be some more that are called as primordial germ cells. PGCs will give rise to primordial germ cells. And these three are primordial germ layers. That is ectoderm, mesoderm, and endoderm. These three would give rise to the somatoplasm, and the primordial germ cells will give rise to the germ plasm. That's how our body is constructed. Some of the amazing things that we need to appreciate is that ectoderm gives rise to certain types of tissues, mesoderm to certain types of tissues, and endoderm to certain types of tissues. If you take the skin, neurons and the melanocytes, which give us skin color, they all are derived from the ectoderm. The brain, spine, the skin. Okay, you can think of it. Brain is kind of very interior into the bones and so on, and skin is the most outermost thing, and they both have formed from the ectoderm. Mesoderm constitutes to majority of the bones, the blood, the muscles, and so on. Right? It kinds of gives the structure and connectivity and coordination between different cells. When I say coordination, it's about muscles. When I'm saying connectivity, it's about blood. And giving a structure to the body is about giving the bones, skeletal system, and so on. At the same time, the endoderm gives rise to tissues or organ systems such as the intestine, thyroids, and I think there are other glands also that are involved that arise from the endoderm, even the lungs and so on. Right? I hope it's not easy to memorize all these things at a time, but we can take a little bit of slowly, gradually. We have to build up our glossary and try to memorize some of these things. Right? You see that, okay, let me take this too. And the germ cells will give rise to either sperm or egg. What I want you to appreciate in this is that there is one egg cell which with a certain type of genome, right? DNA or the genes. And now you're seeing so many diverse cells. The DNA of all these cells is identical, almost identical to the DNA of the zygote they came from. And yet, you see that the the morphology of these cells is so diverse and their functions are so diverse. I would agree that the gametes are not identical to that of the zygote. Why is that? Because firstly, they are haploid, and secondly, or most importantly, is that recombination has occurred. During meiosis, crossing over happens, isn't it? There is exchange of DNA or the genetic information between non-sister chromatids, chiasmata formation, tetrad, and so on. So they are not necessarily identical. Forget about these things, but think of all the other tissues, other cell types. We have about 210 cell types in our body, and all of them have nearly identical DNA as that of the zygote. Zygote itself had a different structure, and now we are having so many cell types, all of them in diverse, morphologically diverse and functionally diverse too. Right? So that is where we discussed in the previous course, epigenetics comes to play, or differential gene expression comes to play. And that is where we will discuss these things in greater detail in Unit 2 or so. Okay, I hope you had a fair enough idea about what we are going to study in the future. And I want you to get used to these terms slowly and gradually. Any questions? I'll stop here. If you have any questions, I would like to take any questions. Okay, if not, we will have a summary quickly, and then I will end the class maybe. You're always welcome to ask any questions. If you're shy to ask now, you can WhatsApp me or even call me up and see if we have something to discuss about it. Okay. So gametogenesis is the production of gametes, and it is spermatogenesis in males, and in females it is oogenesis. In spermatogenesis you have 4 sperms forming, whereas in oogenesis you have only 1 egg cell that forms. 1 egg cell, and the other things, first polar body, which is 2n, and the second polar body, n. Right? And those are the differences. And spermatogenesis and oogenesis. Spermatogenesis happens, say, from the age of puberty until kind of death. The number of viable sperms may reduce, but they are actually produced until a very long time. In oogenesis, there is a limitation. The number of egg cells are thought to be produced already during the embryo stage itself, and it is fixed. And they start producing from puberty until about 45 or say 50 years old, and so on. Beyond that, there is no more ovulation, and the menstruation falls, and that's where menopause starts. In spermatogenesis, meiosis is completed immediately, and it happens during the lifetime of the individual. Whereas oogenesis, the meiosis starts while still at the embryo stage, but only it's completed upon fertilization. Otherwise, it's not completed. And cortical reaction, we have discussed about sperm entry, membrane disruption, and then activation of or influx of sodium ions, or activation of phospholipase C, which will produce IP3 and diacylglycerol, which will stimulate the production of activation of the metabolism in the cytoplasm, and also production or these pouring out or pumping out of calcium ions from smooth endoplasmic reticulum, which stimulates a lot of metabolic reactions for cell cycle to progress. Zona pellucida is the covering that is present on top of the egg, that is proteinaceous or glycoproteinaceous, one that prevents that protects the egg. So we also learned about zygote, cleavage, morula, blastula, gastrula, that is where the primordial germ layers form, and then neurula, neurulation, that's the neuronatal cord forms, and then fetus, where organogenesis and everything else is completed. Primordial germ layers, we learned about ectoderm, mesoderm, and endoderm. They give rise to the whole of somatoplasm. And primordial germ cells will give rise to the germ plasm. These two will make a fertile individual. And you can look at the book Cell Alberts, The Cell by Bruce Alberts, or any other general cell biology books, whatever is there in the early, the developmental biology part, or towards the end of the text is good enough for our purpose. Yes, I'll stop here, and if you have any questions, we can discuss. Any questions?
No, sir.
Okay, so we have 1 second.